So if you miss, please let me know if I cut off.
Yes, sir.
Yeah. Okay. Good morning, everyone. And we were discussing about a chapter that I wanted to take together: analysis of genetic variation and gene expression. And then in that we discussed about different types, possibilities of genetic variations in large scale, small scale, and all. And we have discussed a few of the techniques that are involved. And we will also discuss further a few more today. And for all the techniques, I already told you, there will be about 20 or 30 techniques in the whole of this course, maybe even more. And that in each case, you should know the objectives, principles, limitations, and applications, and then use them appropriately at the correct points. And then I have used these the two two lessons, about genetic variations and gene expression, because expression is about consequences of the variation, right? So it'll be nice to discuss both together. So we also discussed about this problem in the last class. The majority, all of it, was dedicated just to understand this one, and I hope you all understood it. If not, please go through the video and please come up with any questions, and then you can be clear about it. I insist on it because it is likely to come for CAA, a question like this. So to approach all these things, a question like this, you should be able to understand what is given in the question in the first place, all the terms. And second, you should know to interpret these plots. And the third is to relate how a change in one experiment could reflect in the other, and what would that mean, and so on. So keep that focus and try to practice it. And the best possible way is to discuss with multiple friends so that you will understand it better each time. Anybody could just make a gel diagram or create problems, make problems yourselves. If say you are right now, I can see some 11, 12 people, and each of you have made a question by yourselves on trying to imitate these problems or taking it to a higher level, because each of us may be having an expertise in our own way, and try to make a question on it. If somebody is making a question, and if the question is a very good one, that means you must have known the answer, and that means you have learned it. So try to make good questions, something like this, and then probably you'll be in a good state of understanding. Please try to do and post it on the GCR. It will be, people can try to answer as well. Okay, good. So what we are going to learn is a few more techniques which will analyze genetic variations. So genetic variations can happen in multiple ways. Some of them are because of, inversions, deletions, and so on. But if there is a genome, right, in a cell, there is genome. And to start with, all DNA is recombinant DNA, in the sense that DNA, while it is, say, if you are talking about our cells, egg and sperm, then we have fertilization, and during the gametogenesis, there is crossing over or chiasmata formation, where genes are exchanged between non-sister chromatids, and a recombination is made. That is, and then a new person is born with those recombination. What is unique about us is that combination of recombined DNA that we have inherited from the father and mother, and that is what is unique about us, right? There are some genes coming from mother, some from father, and there is the genes actually belong to the parents, right? What is ours is what is unique to us within us is the recombination that has already happened in the parents. That is within us, and nobody else has that. In other senses, apart from this meiotic recombination, there are many other ways things can change. So during replication, there are errors. There are replication errors because of which you may have alterations or mutations, single nucleotide polymorphisms. I mean, substitutions can happen. There is also possibility of insertions, deletions because of slippage. I hope you know what is meant by slippage. If not, note it down. During replication, what is meant by slippage? Either the primer or the template can slip. Assume this as the primer, or template is this. And sometimes hairpin loop, when there are sequences that are repeating during that time, slippage can happen and they can result in insertions and deletions. Substitutions can happen. That is one of the ways of genetic variation. There is another form of genetic variation is it can happen because of viruses. Viruses could integrate, and they can cause large-scale deletion, insertions, and so on. There are again insertion sequences or transposons, and they can hop from one place to another place, and or by at least two mechanisms. One is cut paste, and the other one is copy paste. Okay. So apart from meiotic recombination and chromosomal aberrations that may happen because of cell division during cell division problems during cell division, there are multiple ways how a genome can be altered. You can also have spontaneous ones, in the sense that because of hydrolytic damage and followed by repair mechanisms, there can be a lot of changes as well. We are not going to necessarily, or we have learned many techniques that can detect single nucleotide polymorphisms. Of course, sequencing resolves everything else, right? If we have sequenced the whole genome, we would know everything about it as long as we can analyze it properly. So some of the techniques we will try to learn if we want to have only a specific detail of something. Say, for example, I want to learn about the polymorphism in gene X. There is no point in the sequencing the whole genome for it, because it's too much data for what we are seeking. We can also sequence just the gene itself, only the small part, and try to know what are the genetic variations. Genetic variations do not, by sequencing, for example, if you're doing sequencing, it does not tell, it tells us this DNA sequence, but it does not tell about anything about the consequences. So we also want that. In terms of repair, I just want to highlight this so that you will probably want to find it out. And if you already know, it is fantastic. If you don't remember or recollect, please try to go back to the books and find out what is meant by fail-safe glycosylase. I mean, why I like this is to be able to appreciate how natural selection can be so smart. Okay, how can it be smart? Or probably I should say it as how smart phenomenon can emerge. That is the correct way of putting it, probably, through selection. Okay, so this is a very nice one. If you can Google it up also and find about it. It's fantastic to know about it. It's about repair mechanism. Right now we are discussing something else. But if you have any questions on that, you can come back to me, or you can find other people too. Our textbook is enough actually. Okay, we will learn some techniques on that, and we will try to see how to read them. The first one is about transposons. And I also want to give a brief introduction to transposons. If you can recollect, it's fantastic. If you cannot, that's all right. So I'm going to take a typically there are many types of transposons. I'm taking something very minimal for the sake of understanding. Okay, I prefer Watson book, but you can prefer any of the books that you want. But the point is you understanding the concept. What are transposons? Transposons are something genetic elements, and they have two, say, terminal repeats, or we will just say it as inverted repeats or so. Okay, you can call them as left and right, left terminal inverted repeats, and or LTR, probably L terminal repeats, left terminal repeat, and R terminal repeats. Okay, these are essential sequences that are important for jumping. And here they should be having a transposase gene. Transposase is a recombinase, and it will, upon transcription, it will produce an enzyme that is the transposase. This is the enzyme which recognizes both these repeats, and it is essential in cutting the DNA, cutting the transposon from its donor site, and or it can be also copying ways and then integrating it into a different target sequence. Okay, during recombination, what happens during during integration into a new site? There are some things that happen which we will discuss right now. I hope you understand what is meant by target site duplication. I'm spending time on transposons because we are going to have a paper that we need to discuss about transposition and reprogramming. That's included as a part of your syllabus. So I have taken a sequence, and this is what it is, CGATG. Assume this is the target site where the transposon is now going to jump. And in that case, usually what happens is a cut will be made here and here, say for example, there and there. And then we will have two sequences which will be, I mean, they are separated now. Sorry, here it is C. So we'll have some overhangs like this, CATG, and then we will have ATC, G, C, and so on. So the DNA is cut in this way, and now we have separated like this, and the transposon will settle in between this. I'm not drawing it to scale, or it is smaller in size, but you should understand that these are the LTRs, or the terminal repeats, and there is the transposase gene. Okay. So what happens then is the repair mechanisms of the host will fill these gaps with this TAG. The target sequence, actually, the target sequence has one ATC sequence, whereas upon transposition, what do you see? You have ATC and another ATC. There are two ATC sequences, and that is called as target site duplication. Right? The target sequence is ATC, which is now duplicated. Say if this transposon is moving from here to some other place, what happens to the DNA? So the DNA will become G. So the cut would be made here and here. And what you will have is ATC, and then it will be joined with this A. So another ATC G. CTAG, TAG, C. This is what has formed. You see, these are the signatures. When you find two repeats like this, they are likely, I'm not saying they are always, they are likely that they are signatures where a transposon must have been hit. Okay? And then it must have left. The cut and paste models can be this way. Okay? This is about transposons. So we would want to know suppose transposons are always causing these genetic variations. They can do many things. They can do an insertional inactivation if they they target a gene here, and the product of the gene could be bad. Sometimes, in some cases, the transposon may have a promoter, a strong promoter, because of which the transposon got integrated here, then because of the promoter, it can include the expression of this gene, or it can even reduce the gene expression. So transposons do a lot of genetic variations within the cell. There is no outside influence. It is not about a chemical mutagenesis or physical mutagenesis, or it's nothing to do with meiotic recombination. All we are looking at here is within the genome, everything is there, and it's kind of, you can say it as a biological mutagen, you can call it. And it can do that. Of course, within the cell, majority of the transposons are suppressed by methylation and so on, which we will discuss a few things about DNA methylation in a later class. I hope you have good enough introduction to transposons, and then if we want to analyze it, when we want to analyze, typically we would want to know how many copies are there of a transposon. Then we can just go by RFLP, restriction length fragment polymorphism. And if we have done, RFLP includes restriction digestion, isolation of DNA, restriction digestion, and then blotting. Then we add probe. So because we want to know the copies of the insertion sequence or any transposon, then the probe should be homologous or complementary to the transposon. And then we can, if there are two, we would usually get two bands. Whether we get two bands or not is a difficult one to explain. If you get two bands, the most accurate sentence that I can make out of it is there are at least two copies of transposons. This can anybody tell me if there is upon transposition, is there a possibility for transposon to give the same fragment size even after transposon?
Sir, could you repeat that question? I'm sorry, not able to get it properly.
Yes. Is there a possibility for, say, there is a insertion sequence or transposon which has also hopped on to copy paste to somewhere else? Okay. Then is it possible to get the same fragment size? Say, for example, 5 kb. And this is, can this also be 5 kb? I'm just trying to illustrate that. Say, for example, there is a target site, new target site here. This is the transposon that is labeling as 3 kb, just for sake of understanding. This is 1 kb, and here is another 1 kb. So total of The arrow marks here indicate the restriction site. Okay. And now I have a 2 kb target site, and the transposon has copied and pasted here. So the genome totally has the one that it already exists is there, and in a new place too, it has moved to a new place as well. Right? You have 2 copies of transposons. The size of it is 5 kb. From here to here is also 5 kb. How many bands would you expect on after southern blotting?
Only one, sir.
Yes,
One, sir.
it will be only one, right? So we have to think about that as well. But otherwise, in a simplistic way, if you find 2 bands, then we can say that there are at least 2 copies of transposons in 2 different locations. Or yeah, that is what you can say, 2 different locations. Okay. So here the experiment is usually done. I think this is about insertion sequence, which would imply, not necessarily, but imply that it could be in bacteria. So in bacteria, if you want to study, you can check this out easily like that. Okay. Good. So this is simple way of doing. All we have changed here is about the probe. We made the probe specific to the insertion sequence that we are interested to analyze, and thereby we can estimate or guess estimate the number of copies. There is another one called as variable number tandem repeats in our cells, in our genomes, and different people have different copies of those. There are several repeats, which could be from base pairs to kilobases sometimes. And actually, they're small, 100 base pairs or so approximately. And these are regions which can keep increasing or decreasing in size. So in this case, there are 2 copies of the VNTR, whereas in here we have 4 copies, and they could be changing. So if we know the region where these VNTRs are present, and we want to find out we want to know how many repeats are present, then it is simple. We have to design primers in such a way the unique sequence, this part of it is unique, right? And only this part of it is the repeat part. So we could use those, and by designing primers like this, I think you can see that this one, the first copy will, because it has 3 copies, each assume is 1 kb. So it will give a 3 kb fragment, right? Approximately. And if it has more, like 4 copies, it will give a 4 kb fragment and a 5 kb fragment in this case, if it is 5 copies. And by doing this in just PCR products and electrophoresis, you can guess the number of copies that are present. Okay. So as I said, it is important that you try to read about what is meant by slippage with regard to replication errors. And this becomes relevant, how these number of copies could vary. You try to read upon it, and then we will discuss if required. But as a technique, and that's sufficient for the course, is it is simple PCR primers, and how you analyze it is the important part here. That is how you can test. You should, okay, there is a question I'll have. What would happen if I have designed primers for VNTR? What would happen then? Anybody? Just as a complication, I would want to know what you think will happen. The arrowheads here indicate the primer regions, right? Instead, I'm choosing these, I'm designing primers for the VNTR, then what will happen? If it can bind here, yes, please try.
So it will be able to, as you were about to say, I guess, it will be able to bind with all the VNTRs, all the copies of it. So you'll get
Yes.
1 KB fragments, but the intensity would vary. So maybe we could look into the intensity and find out the number of copies.
Why would the intensity vary?
Because there'll be 4 1 KB fragments or 5 1 KB fragments.
Okay. What is the limitation for PCR or intensity? That is also going to become relevant in the future classes, so I wouldn't mind discussing that. Intensity of the band is dependent on what? On the DNA that is formed? Yes?
Yes, sir.
Okay. And that is dependent on? That is dependent on the
Template.
replication, right? And the replication is dependent on template. Yes. Then what else? DNA polymerase.
Polymerase.
That's right. DNTPs and buffer and all, right? If you do not have the, and there is one more thing.
Primer.
No, primers we already assume. Yes, okay, primers are there. Number of cycles. Okay, in a traditional PCR, I think you have to see the RT-PCR, real-time PCR, differently from the traditional PCR. Template is essential, only then replication will happen. Polymerase, all of these things are essential for replication to happen. Which one is the limiting factor is the for intensity? That is what we are focusing. Okay, for intensity, which one is the limiting factor? In a traditional PCR, template is not.
The template
is not. Template is required, but it's not necessarily the limiting factor. Because
so that's in the number of cycles or the polymerase activity. Right?
Yes, because we are typically running these cycles for between 25 to 30 cycles. Okay. And so that means we are exhausting all the DNTPs primers available. Maybe after, if you calculate the number of products that can form, it will be billions. And at the one point, you will have the exhaustion of DNTPs or maybe polymerase, because of which, even if you have 2 templates or 4 templates, they both will form same intensity bands if you run all the cycles. In real-time PCR, which I think we'll discuss in the next class, so it will become relevant there. If we had only number of cycles is only 5 and then check, maybe then you can probably, it will be proportional to the template. But if you have exhausted the whole DNTPs and ran the full cycles like this, then usually the band will not, the intensity is not representing the concentration of the template. Okay, I'll just discuss this again at this point. You can keep it open and think about it and have a look at it also, and then we can discuss this further when we take up RT-PCR. I think we'll discuss in the next class. Okay, but what can you expect is the question I had was, this can give one product, again this one can give another product, and this one can give another product. There are also possibilities that this one and this one can give a product if we add in the same gel, same PCR mixture, but it's all going to be a messy one. We will not be able to see something very clear. There will be more uncertainty about or confusion in the way we can interpret the results. Instead, if we have taken the borders of the genome from where the VNTRs actually start and end, then we can get results something like this. Based on their molecular weights or sizes, we can determine how many copies of the VNTR it has, the genome has. Okay? So there is also another one, which you call it as single strand conformation polymorphism. So basically, this is used for you can use it for multiple purposes. If whether you want to find out if there is a polymorphism or not. I like this technique because it involves some biology thinking and all. So what we do is you make, say, for example, this is sample A and here is sample B. You took a radioactive or radio-labeled primer, N-labeled. We will discuss about N-labeling and random labeling at later time. It'll be good if you can already have a look at it and come back. So we have two samples, sample A and sample B. We took the genome and added only single strand and tried to produce the complementary strand of it. Both are labeled. And then you denature them. This is denaturation. So you have here double-stranded ones, which are now single-stranded. And you cool it rapidly. Rapid cooling. So normally, if you're going to do slow cooling, it will start base pairing like this. But if you are forcing it rapid cooling by putting in very cold temperature, so these are two strands, which if it is slow cooling could become like that. If it were rapid cooling, the bases here can start to base pair among themselves and forming hairpin loops and so on, and forming different structures. And that structure, that is the conformation it has is dependent. The conformation of proteins or nucleic acid, say for example RNA, they are dependent on the sequence. There is a stable conformation it will form at different conditions. Here we are separated them, denatured them, and rapidly cooled them. And in those conditions, they have a conformation which is a function of their sequence. They will form the way the sequences are. If there is a variation in the sequence, okay, it could be a single nucleotide polymorphism, or it could be a little bit small insertions and deletions, a few of them, then there is a variation in the sequence because of which the conformation could be altered. And now when we put it into, when we run the gel, based on their conformation, they run differently. Okay, say for example, I will try to make up a gel picture for you, and if you can interpret it, then we have everything good. I'm taking samples X, Y, Z, and P. Assume I have done the same SSCP analysis, and then I have a band like this. I got bands like this. Which one is the largest of them in size?
X.
Which one is the largest?
The first one's X, sir.
Why? Why is it the largest? Anybody? I also encourage others to speak up. I don't want to call names here. It will be nice if you can try.
So I guess it depends on the diffusion of
It is
because the conformation,
electrophoresis. Yes. Yeah, tell me. Continue.
like because of the conformation, like if it is large, then it might not be able to penetrate well. Something like that.
Okay, I want to go back a step in electrophoresis. I have isolated plasmid. I have isolated plasmid, and then, sorry, then I have run it on a gel. Okay, it's coming wrong. And assume that I have 3 different samples. And this is mostly recollecting, trying to help you recollect what you have learned in genetic engineering, maybe. This is the plasmid, freshly taken, isolated one. This is plasmid that was nicked plasmid. And this is double-strand break plasmid with the double-strand break. Say, assume there was a single restriction site, we cut it with it. So if I had performed electrophoresis, how would the bands look like?
The plasmid would be closest to the well.
This is the well. Okay,
Yes.
tell me. Okay, yes, tell me, please. It'll be, all of them are plasmid, the same plasmid, without any treatment, with a single nick and with a single double-strand break. Yes. Yes, please, go ahead.
So the plasmid would be closest to the well, followed by the
Here?
next one.
Followed,
Yeah,
this
in a
is,
way,
yes,
just
tell
the
me,
order.
please.
Plasmid, then
This
the
way,
next plasmid, then the double-stranded one.
here,
Yeah.
and here?
Yes.
Is that what you meant?
Yes, sir.
Okay, why is that?
Because the double-stranded broken plasmid, that will be linear, and therefore it can travel faster. It can
Okay,
diffuse faster.
okay,
And a plasmid is totally circular, so it will have more space, and it will not be able to traverse through the gel. So that would be the least traveled from the well.
okay. Have you learned about supercoiling?
Yes, sir.
Yes, then yeah, tell me, what do you know of it? Which one do you think will go pass through? Assume this is a pore. Okay, this is the pore I made. And then I'm talking about a DNA that is like this, a DNA that is like this, and a DNA that is like this. Which one can pass through the pore easily?
The last one.
This one, right? This is supercoiled. When it is supercoiled, the plasmid, majority of the DNA in generally, normally, okay, is negatively supercoiled, because of which it forms a tightly packed up thing like this. This one can travel through the pore faster. Okay, so it will actually be the reverse order. Okay, but the one that is, I think the linear one travels somewhere here. If that is the linear one, then the supercoiled one will be somewhere. Oops, sorry. Supercoiled one usually will run faster. I think, I hope you know about covalently closed circular DNA, right? CCC, that is completely supercoiled, usually supercoiled. When it is in the bacterial cell, you isolated it, it's usually supercoiled. And there is also open circular DNA. This is where the nick, there is a single break that is in relaxed state. That means there is no supercoiling. Supercoiling is a type of potential energy. And then we have the double-strand linear DNA, where we have this one. I would want you to have a look at these. All of these are of same size, right? Molecular size wise, they're all same. What is differing is their conformation. Based on this way they are organized within themselves, their mobility through the pores will be different. Okay. Size could be, if all of these are linear DNA, say for example, if we take 3 different sizes and all of them are linear DNA and we run through the gel, then they all will travel based on their molecular weight alone or the size alone. But if they are something like these with plasmids and so on, which there is the possibility of being supercoiled, then supercoiling will result in conformations, and those conformations can also influence the mobility of the DNA through the agarose gel. Okay? Please look up on it, and we can have any discussions if you have a question on that. So the same thing applies here. Here we are trying to analyze based on conformation. We have rapidly cooled them to make intramolecular base pairing, and based on their sequence, they would have attained a conformation. And if there is a variation in the sequence, then the conformation would be different. If there is a difference in conformation, then their mobility will be different. If we had the same size of both, assume it's the same gene but different alleles of it, assume the size in terms of base pairs is same, then their mobility, if we followed these steps, the mobility will be dependent on their conformation. So if we did like this, X does not mean it is larger in size. It is what we can say is that all these 3 have differences in sequence. We don't know what is the difference. All we can say is there is a difference or variation. We can also say there is likely that these 2 are maybe having the same type of, they are identical or very, very similar or most similar. That is what we can say. I hope you also understand that I'm not saying this is what they are identical. I'm saying they could be, or they're most identical or most similar. Okay. So keep that in mind. About conformation can also influence the electrophoretic mobility, but it depends on the molecule, how you have made it. Yes?
Yes, sir.
Okay, so this is, yes, you have a question? Anybody?
No doubt, sir.
Okay.
No, sir.
Okay. So this is how usually the SSCP would look like. Based on that, people try to find out. Say, in many times we want to, before going for, say, now sequencing is very easy and kind of affordable. The previous times it was difficult, and there was not as much of consultancy like outsourcing possibilities. So they would have used different screening methods to see if there is variation. And if you think there is variation in, as you might have found here, then they would have tried for going for sequencing and to know the nature of the sequencing. And of course, we still have to do expression analysis and find out the consequence of the sequencing. We may know many things about it, but we have to prove that the consequences of the genetic variation. Okay. So I will introduce a few of the things. There are, in different labs, you will find different expertise and different approaches. Say, if there is a bush and there is a bird in it. Sorry. There is a bush and there is some noise coming out, and you are the hunter. okay, and you don't know what to do. What you would do if you had is you would have put a stick there and just try to beat the bushes. That is what you call it, right? Beating the bushes. Beating the bushes. That means you don't have any certain idea that there is something. It could be a snake, it could be a bird, it could be something that you could use it for some purpose. All you know is there is some noise coming and you're very hungry. Then you just do it and something may pop up and you might probably take it or catch it. That is the beating bushes approach. But you should have had some indications that there is something different about it. There is another way of seeing it. You have a tree and tree has a branch and on that there is a bird. You take an approach where you would take a gun or so and you will shoot the bird only, right? When we do things, when we don't know anything, this is a good strategy. When we don't know, all we know is faint noise. That means there is something there, but we don't know anything. Then it is a difficult situation. We don't know what to do also. We don't know what we are doing is right or wrong. But we have to do something about it because we have a need. Right? And then some labs have expertise in trying to beat up the bushes and see whatever comes up and then work on it. Or they will beat up the bushes and see that, hey, I beat up this bush and these are the things that came, and they could publish that. And different other labs could take up and read it in detail. There are other labs which are highly specific. They might be working on a certain phenotype, certain gene or protein and so on. Then they are interested in that particular gene. They will give full knowledge about one gene or one character. So we'll just write one gene, right? In the genome-wide comparisons, this is much more this is holistic and this is reductionist. Both are important, okay? You cannot, in holistic one, you don't get fine details about what a particular gene is doing. And in reductionist approach, you cannot really put, just knowing one gene doesn't mean that you know the whole cell or what is it doing within the cell. Both are important. And so this is a difficult one, holistic one, but whether how much is useful is dependent on how much we are going to study about it. But they can give us an overall picture or importance of that gene of the whole system. Okay, this is trying to beat the whole system and see what all, because we have some indications that there is a probability, there is a problem or so. And then it will give us the overall picture of what are all the genes that could possibly be involved in making that noise. Whereas here, if these people can take over these hits, and then they can work on it further to give very fine details about each of the particular genes. Okay, here we are trying to see something about genome-wide comparisons. In the previous ones, whatever we discussed, they are usually specific to certain locus of a genome or a certain gene and so on. If you want to study that, the certain gene or specific ones, the previous techniques are useful. But you can also do these overall or this holistic or genome-wide comparisons. This is using microarray technique, and I'm going to mix it up again with a little bit because genome-wide again can be in whether we are doing it at the genome level or transcriptome or proteome. Okay. because this is going to come, these are always related. So a chip is something like this, which is a glass slide. And on the glass slide, I think I have a picture. You will have an array of, you call it arrays. These, each dot there indicates, each small square has multiple dots made by robots usually, and each of them are tagged with a particular sequence. Okay, so one dot, one square probably, in one dot, you will have several single-stranded DNA like this, representing one gene. Okay, and this could be representing another gene. So the oligo is highly specific for that one gene, and say gene X, and here it is for gene Y. This could be for gene A, and so on. So on this glass slide, these are printed. And you have, that means you can cover so many genes of it. This array will have, which is about the size of your thumbnail or so. Okay. That is a significant step forward when this technology first came. And you can analyze many things using this microarray technology. And imagine the approach for this is something like this. You are comparing two different cells, cell A, or you can say organism A and organism B. Same species, but there are variations. So you want to know what are the variations, and then you can do these things. The design of the microarray will depend on what you are analyzing, and it's mostly what is put here is the genomic, the genes, the open reading frames. In majority of them, they would have, because we have bioinformatic tools, annotation is done, and then they would have identified an oligo that is specific to it, and then they would have printed it there. That means usually it is only about open reading frames. It's not there might be missing the have a gene here and here. You have you know that it produces a protein and it's a gene, and it is so we can design an oligo and spot it here. For the other gene, we can spot here. What about these sequences? These are the intergenic regions. They are not necessarily showing up here. Of course, there are variations to this, but we are not going to discuss at this time. So you can take two different organisms as DNA. You can isolate the DNA from both of them. And okay, I'm running out of time, but okay, I'll go here. So there is a protocol where, say, each one is representing one gene. Right? And we know we have different ways of how the combinations can be done, color combinations. Red and green gives yellow, the primary colors, and then the secondary colors, and so on. So what we have here is we are going to mix cells from two different organisms. I have a protocol. I will give an introduction, and then maybe we'll discuss in the next class. So here is a test, like some organism A, and here is the organism B reference that we know is normal. That is the reference. The test is whatever problematic or so we are interested in. Here they're not using DNA directly. They're using cDNA instead. They would have reverse transcribed, and then you would have put it into fluorophores. One of this is with, say, for example, red, and the other one is colored with or tagged with green fluorophores. And if this is a spot, and each of these oligos are here, okay, I should use a different color. I'll stop within 2 minutes. Just give me a minute. Yeah. Here are the oligos that are already attached. And now we may have some of them coming from the test reference and binding here. If only the reference bound in a particular one, then when the laser is hit, it will give green fluorescence. If there was only red binding or no reference ones are binding here, then it would have given red. But if we have green as well as red also base pairing here, then it would give both red and as yellow ones. And there is a powerful microscope to read all the fluorescence from all the spots, and then it will analyze the color, which ones are red, which ones are green, and which ones are yellow. Red means it is present in the reference, but test does not have it. If it is green, that means test organism has that sequence, but reference does not have. If it is yellow, both reference and test have it. And if there is no color, neither of them have it. Okay? If you have any questions, please ask, but otherwise I will try to repeat this in the next class too. Any questions?
So this is based on the initial transpose on concept that you've been teaching initially.
Okay.
So in the first part of it, like there is a single band, but you mentioned
this one or this one?
This one, sir.
Okay.
With the
yes.
band.
Yes.
So in case one, you mentioned that there is a possibility of the transposon being in a single site, but you also elaborated on how it could also mean that there are two different sites in
Yes.
which this band is present, but it could give a same it could give a
That's
single
right.
band.
Yes.
So how do we basically understand which one is the case?
No, it's not. It's difficult to be certain about it. We cannot be certain about it. But usually it will be multiple things. Multiple probes can be made, and if it is just about insertion sequences, it cannot be set very easily. The one variation that you could do is, say for example, I have done it with eco R1. Okay, I'll just put it as eco R1. And then there is a possibility that both would have given, say, 5 kb, 5 kb like this. Right? Do you agree with that?
Yes, sir.
There is a possibility. Then I could change the restriction enzyme to BAMH1. It's highly unlikely that both would have given the same fragment size. Did you understand that?
Yes, sir.
Yeah, so we can play around with different restriction endonucleases, which thereby if you are getting we are likely to get different fragment sizes, and that is how we can figure it out. That's one way of doing things.
Thank you, sir.
Did you understand the?
Yes, sir. So like
Okay. Yes.
when do we stop doing that? Because there are multiple possibilities.
We don't stop doing. I mean, usually when we do research in papers we want to publish, people try to show something in 3 different ways: in vitro, in vivo, or say using this technique, using that using a different technique, and so on. So probably we'll have to do it with different. I mean, if we don't have to do too many stuff, we just change it H1. Okay, and then we will be able to figure out if it is same copy or not, because it's almost near impossible that it would give eco R1 might have given the same fragment size, and it is that BAMH1 also will give the same band size is very improbable. Very improbable. I'm not saying impossible, it's highly improbable. So just testing with the different restriction endonuclease and doing the same southern blotting would be sufficient actually. If not 2, 3. they add one more or try it with a different the third restriction endonuclease. That should be sufficient.
Thank you,
Yes.
sir. Yes.
Yes. Okay. Thank you, everyone. Yes.
I'm so sorry, sir. I have a couple of doubts.
Okay.
Um,
The others, if you want to leave, you can leave. You can stay if you want. Yes. Parvati.
so with respect to the variable number of tandem repeats,
Yes.
you mentioned that if we design the primer to be within the tandem repeat itself,
Yes.
then it should be able to bind with every other repeat. Is
Yes.
that right?
That's right.
Okay.
Yes.
So I'm still not able to get the point why the intensity would not matter. So,
Why intensity would not matter? Okay. Then I will have to go to. I must be having it here. That is where I was supposed to teach. Yes. You know about real-time PCR. You must have read it, and but it's all right. We will discuss this here. Which one do you think is having higher number of templates to start with? This is the product signal coming from the product that is being formed after each round of PCR cycle.
is that A, sir?
A has more, right?
Yeah.
More than B, and B has more than C, right?
Yes, sir. Huh.
More template, little lesser, and this is even lesser. What is here? What are you seeing here? After these many rounds of PCR cycles, this would reach saturation very fast, where there would have been some, what do you call it, some limiting factor because of primers or template or DNTP and other things, not necessarily about the template alone. If you have stopped the reaction here at this stage and you analyzed, then it is proportional. The fragment formation, the product formation is proportional to that of the template. But at this stage, the fragment, the product formation is not proportional to the template because it has saturated. It stopped further polymerization. Okay.
Okay. Yes, but we can still interpret this using the number of cycles it has taken to reach the saturation, right? We will still have
Yeah,
that.
that is called as real-time PCR. There is a technique, we will learn about it, and then probably we'll get it much more clarity on that. Okay.
Sure,
Real
sir.
time. What does real time mean here? After
We look
each
into
round of PCR, we are testing how much of product has formed. While the PCR is happening, in real time, after each round, we are testing how much of product has formed. In traditional PCR, we perform the whole PCR and then we analyze. Right? This is after PCR analysis. In real-time PCR, while PCR is happening, we are testing the amount of product forming. That is real-time PCR. So at that time, the product formation, if we will know based on this a graph that we generate like this, we will know which ones had more template and which ones have less. Yes.
Thank you.
Anyway, in the
Yes.
next class we will discuss on that. Yes.
And one
Any
last.
other? Yes. Go ahead.
So you mentioned that in order to prevent this condition, we can design the primer such that it is out of the tandem repeats itself, like in the
Yes.
beginning and end of it. But
Right.
how do we know the beginning and the end of it, sir? Until
Yeah.
unless we have the sequence.
Yes, we wouldn't know that. We wouldn't know that directly. What we know is, say, for example, if one person we have identified VNTR in one person's, and then this locus could be present in all of us, and we don't know how much this VNTR could be different in different people. And I think this is very well used in forensics and other stuff as well. Different people may have different, RAPD analysis and so on, probably we will discuss those at the later time. But for this, to get the initial information, we would have needed some form of, would we even design the primers, right? So we need a prior information, and based on that, we can see how people are varying in terms of the repeats, the number of copies they had. This usually does come from sequencing.
Thanks for
Yes?
the clarification, sir.
Yes, okay, good. Thank you, everyone. Thank you for attending and staying. Have fun.
Thank you, sir.
Yeah, bye-bye.